My name is Adam Salmon. I'm the co-director of the San Antonio Aging Shock Center. In this talk, I'm going to provide an overview of how we view the role of the Shock Center in providing service to the field of aging. I'm going to give an outline of our research cores and the services that they provide. And lastly, I'm going to give an example of the type of assistance our Shock Center can provide to investigators to help enhance their transformative aging research. Overall, we view the San Antonio Shock Center as a horizontally integrated, enhanced platform to serve investigators in their transformative biology of aging studies. And what we mean by this is essentially we would like the San Antonio Shock Center to be a one-stop shop for investigators looking at mammalian aging research. And in this, we can provide assistance from the point of the research idea of an interested investigator through consultation with experts in aging that we have at our San Antonio Aging Shock Center. We can provide assistance through each of our research cores, which I will talk about briefly. And what we view that this assistance throughout in this horizontally integrated program can do is accelerate the research of investigators so that we can help assist them in developing their transformative aging research discoveries, to help assist them in high-impact publications, as well as provide assistance with both grant support from the Shock Center, as well as assist them in getting their own independent grant support for their research. We have 6 interrelated research cores. As with all Shock Centers, we have an administrative core. The directors are Randy Strong, Peter Hornsby, and myself. Our overall goal is to monitor, stimulate, sustain, evaluate, and report progress of the San Antonio Aging Shock Center. We also provide outreach for aging research through various programs. Our main program is our annual Bar Shop Symposium on Aging Research. This year, this will be a virtual symposium on immunology and aging for anyone interested, and this happens every year in October. We also provide the networking and interaction with other Nathan Shock Centers, as all our Shock Center administrative cores do. In addition, through our statistical expertise with Jonathan Galfond, we can provide statistical assistance for research projects both before and after their projects are performed. Again, like all Shock Centers, we also have a research development core led by Jim Lechleiter and Peter Hornsby. We have 3 sort of ways in which our research development core can assist investigators. The first is through external projects. These are available to all investigators, both inside and outside of our institution. We also have a set of internal pilots supported by administrative funds, institutional funds. These are open to our local investigators and provide support for junior faculty and aging research. Our research development core also has a career development role in which we provide a mock grant review process as well as design assistance, and this is open to all investigators externally. And in fact, is part of our pilot funding program. Our next research core is the Aging Animal Models and Longevity Assessment core. This is led by James Nelson and myself. In this core, we provide access to 5 models of interest to biogerontology: mice, rats, naked mole rats, the Marland mole rats, and marmosets. Now, obviously, mice and rats, the rationale for using those for aging research is clear. For those that don't know, we also keep a colony of naked mole rats and the Marland mole rats, which are long-lived rodent species that have developed many transformative ideas about aging research. The marmoset is a non-human primate, and we maintain a colony of these small animals for aging research. Now, for all 5 of these species, we can provide animals, tissues, and cells to interested investigators. We can also, within this aging animal core, we can provide longevity assessment of genetic mutant mouse models, rodent models, dietary interventions, or pharmaceutical interventions. We can provide assistance from the point of breeding to complete lifespan studies. And we also, in the animal core, provide all manner of training, including design consultation, and assistance with grants, and manuscript preparation. The integrated physiology of aging core expands on the aging animals core to actually measure functional changes with age in mice, rats, naked mole rats, and Damara rodents. This core is led by Nick Musi, Veronica Galvan, and Elizabeth Fernandez. Essentially, we can provide a whole suite of health span assessments within this core. We've essentially separated them into 3 different types of assessment: metabolism and bioenergetics, including glucose metabolism, respiration, mitochondrial function. We can also perform neuromuscular and cardiac function, including muscle functions, balance and coordination, locomotion, echocardiography, and frailty assessment. And lastly, we can provide cognitive and behavioral assessment of aging animal models, including working memory, spatial memory, and novel object recognition, and brain vascular. Our next research core is the Analytical Pharmacology and Drug Evaluation core, led by Marty Javers, Brett Ginsberg, Marissa Lopez Bizan. The basic idea of this core is that the Analytical Pharmacology and Drug Evaluation core can measure in blood, in solution, in food preparation, any type of drug or natural product through mass spec and HPLC for intervention type studies. And so pharmaceutical aging interventions, they can assist with design of dosing studies, how best to get that drug into your animal model. They can assist with integration analysis, looking at pharmacokinetics and pharmacodynamics. And they can also assist with interpretation of those studies along the way. Our next research core is the Pathology core, led by Yuji Aquino and Gene Hubbard. And both Drs. Aquino and Hubbard have had years of experience assessing pathology in rodents and non-human primates. And they can provide service, again, through all manners of different types of service, including end-of-life pathology or cross-sectional studies. They can also assist with histological analysis or even something like your pathological archive. And this core also maintains a pathology archive for interested investigators for their own studies. The basic idea is that by addressing all of these types of studies, they can look at the pathological mechanisms of aging and how they drive the aging process. So an example of how the integrated research cores of our Shock Center can assist an interested investigator is presented here with an external pilot that was funded in 2019 to William Jack from the Scripps Research Institute. And so his study was titled Fluspirulline as a Modulator of Metabolism. And so his preliminary studies that he presented to our Shock Center as rationale for his external pilot were based on a cell-based assay to identify compounds that mimic the translational state shift associated with longevity intervention. And with this cell-based assay, this high-throughput assay, he identified fluspirulline as a novel mediator of translation. This is interesting because this drug is an FDA-approved antipsychotic used in schizophrenia. Now, in his preliminary studies in vertebrates, he had found that both in C. elegans and in Drosophila, fluspirulline can extend lifespan in these model organisms. Now, this was the rationale for the interest in testing whether this might be an intervention that would be of use in mammalian studies to extend longevity or improve health. And that was the basic rationale for the study. Now, Bill had not at the time worked with rodent studies, so it was essentially, he worked with consultation with our dean and admin cores, with the collaboration, the development, and the coordination of the project design, and essentially worked with these cores to figure out what would be the best outcomes to assess that our Shock Center could assist. In addition, obviously the funding for this project came through our RD. The Pharmacology core then stepped in and was able to help with developing assays of fluspirulline in solution in the rodent diet prepared for the studies, as well as in animal blood. Along with this Pharmacology core was able to do pharmacokinetic curves in male and female mice to determine whether there were sex differences, how often this drug should be dosed, not how often, but what the concentration of the drug should be used for the dosing, This was done in concert with the Animal Corps, which was able to purchase the animals for a bill to provide the care and the maintenance of the mice, provide basic orthometrics along the way, the body weight composition, food consumption, as well as sample collection. The IPAC has been able to assist with sample collection. And COVID-19 has slowed some of these studies, but the goal is to then look at metabolic and mitochondrial assessment of treatment with fluspirulline, as well as the effects of this drug on locomotion, cognition, and stress-related behavior. So, he has not yet used the pathology cohort because the animals were not sacrificed at this point. But essentially, this project has used almost all of our cores at the Shock Center to develop the pilot studies from an investigator who did not have experience in intervention studies in mammals, to now provide them with these basic pilot studies for their transformative aging research. So overall, with San Antonio Nathan Shock Center, our big goal with this study is to figure out, with this story, is to figure out how we can assist you with your biology of aging research. And what I hope to take home of this is that we provide what we view as a one-stop shop approach to mammalian aging research, especially interventional studies, looking at drugs or compounds and how they affect mammalian physiology. And we can assist with all these aspects of an interventional longevity study, from the point of, like the previous study I just showed you, not knowing what the pharmacokinetics of these drugs are in mice, to the complete longevity study. The San Antonio Shock Center can also provide each of these services as an a la carte approach. If you just want pathology or just want longevity studies, we can provide. In addition, we view that we can provide innovation and functional and physiological assessment of mammalian aging across multiple different animal species. And so if there is anything that the Nathan Shock Center can do for you, please reach out for assistance. And I've put our web address here. All of our core leaders are available on this web address, and please reach out as necessary. Thank you for your time.